Hi guys, this is Ms. Romani, and for today's lesson we're going to be learning about chemistry. More specifically, we will be reviewing some of what you learned about elements and atoms in your past chemistry classes. Now, you're studying biology, which, as you know, is the science of life. But in order to fully understand biology, you need to understand chemistry, because living systems are essentially chemical systems. Now, this is particularly important for this grade 12 biology course, which focuses mainly on biochemistry. Even the simple act of eating an apple is all about chemistry. The apple is made up of molecules of carbohydrates or sugar called starches, which the body will digest via chemical reactions. In the digestive tract, the starches will be broken down into smaller carbohydrates, and then when those sugars reach the cells, they will undergo a series of chemical reactions that will transfer the energy in sugar bonds into a more usable form. This molecule, called ATP, will in turn undergo chemical changes in order to drive the cellular processes that keep a body alive. So in the end, biology is just chemistry. Organisms are basically made of elements and compounds, which are made of atoms. So there are almost 120 different elements that we know about. Each element has a symbol, usually the first letter or two of its name, and they can be organized in a periodic table based on their physical and chemical properties. Elements can combine with each other to form compounds. For example, the element sodium, a very reactive alkali metal, can combine with the element chlorine, a very poisonous nonmetal. When these two highly reactive and dangerous elements combine, they form the extremely stable and harmless compound called salt, or sodium chloride. The process of combining those two elements is actually called a chemical reaction. In this particular case, a very reactive one. In this experiment, the reaction of sodium with chlorine was actually conducted underneath a mesh filled with unsalted popcorn. Much of the salt produced was expelled into the air by the force of the reaction and salted the popcorn, which the experimenters then ate. Crazy. Out of all the elements in the universe, just 4, carbon, oxygen, hydrogen, and nitrogen, make up about 96% of living matter. The remaining 4% is just 7 other elements and only trace amounts of a few others. And as you may remember, an element is a substance that is made entirely of one type of atom. Atoms are the building blocks of matter, and they are unimaginably small. For example, an adult human is made up of about 30 trillion human cells, and each cell is made up of approximately 100 trillion atoms, most of them carbon. So if my math is correct, that means that there are about 3,000 trillion atoms in a human body, which is actually 3 quadrillion atoms. A number so big, it is hard to imagine. So yes, atoms are unimaginably small, but that doesn't mean that we don't know what they're made of. Almost all of the mass of the atom is found in a dense core called the nucleus. The nucleus is made up of positively charged protons and other particles called neutrons that have no charge at all. Orbiting outside the nucleus, way in the distance, are negatively charged electrons, which have very little mass. I've been using the term charge, but what does that mean? What do you need to know about it? Well, you don't actually need to know very much. Charge is fundamentally a property of certain particles, like protons and electrons. What you need to know as a biology student is that protons have a positive charge, electrons have a negative charge, and that those two charges cancel each other out, so that an atom with an equal number of protons as electrons will have a net charge of 0. You also need to know that like charges repel and that opposite charges attract. In this representation of a carbon atom, the atom is shown as a sort of miniature solar system with the electrons orbiting around the nucleus. Now in chemistry class you will have learned that the reality of atoms is much more complex. The region where the electrons are found has 3 names. They are called orbitals because electrons kind of do orbit around the nucleus. They can be called electron shells, or they can also be called energy levels, because the amount of energy in electrons corresponds to how far away they are from the nucleus. This is actually a concept that we will revisit during the metabolic processes unit, when you will learn more about how electrons gaining and releasing energy can drive cellular processes. Rather than following a predictable orbit or path around the nucleus, electrons form more of a cloud of negative charge around it. And because opposite charges attract, it is the attraction that the negative electrons feel towards the positive nucleus that keeps the electrons moving around it. In an uncharged atom, the number of protons is equal to the number of electrons, which makes the atom neutral. The number is usually about the same as the number of protons, but it can vary. We'll learn in a bit what happens when the number of electrons is not equal to the number of protons in an atom. We will also learn about what it means to have variation in the number of neutrons. All atoms with the same number of protons are considered to be the same element. So all atoms of the element carbon have 6 protons. Each element has its own 1 or 2 letter chemical symbol, usually the first letter of its name in English, but not always, since the languages used to determine the chemical symbols was originally Latin or Greek. The chemical symbol is listed in a square in the periodic table of elements. That square will also tell you the atomic number for that element and the average atomic mass. The atomic number and atomic mass can give you a lot of information about a particular element. The atomic number is equal to the number of protons in atoms of the element, and since in neutral atoms the number of protons is equal to the number of electrons, then it kind of indirectly also tells you the number of electrons. It also gives you the average atomic mass, which is the average weight of an atom of that element. And since electrons have virtually no mass, and since each proton and neutron have an equal mass of 1 atomic mass unit, this number is basically telling you the number of protons and the average number of neutrons in atoms of that element. Now you've probably noticed that this number is often a decimal, and that's because the number of neutrons is not always the same. When representing this information, scientists often use the form called standard nuclear notation. Now notice that in this example, the given mass is not a decimal, and that is because right now we are looking at the information for a particular isotope of carbon, not all members of that element. In this particular example, the carbon atoms have 6 protons and 6 electrons and 6 neutrons. We can calculate the number of neutrons by subtracting the atomic number from the mass number. So what is an isotope then? We'll get to that in a second, but first let me explain something about the nucleus. Now we learned earlier that opposite charges attract, and that it is this attraction that keeps the electrons moving around the nucleus. But we also learned that same charges repel. So what's keeping all these positively charged protons together in the compact nucleus of an atom? The answer is a nuclear glue in the form of neutrons. Neutrons prevent the positive charges of protons from repelling each other, thanks to something called strong nuclear force. Like they literally named it strong force, probably because it's almost too powerful to imagine. Much stronger than gravity. So the neutrons hold the atomic nucleus together, but their numbers can actually vary. Elements can exist with slightly different number of neutrons, and we call these isotopes of an element. There's often more than one isotope for each element. Sometimes there are isotopes with not enough neutrons in a nucleus to hold it together, or with too many for it to be stable. Isotopes that have unstable nuclei are known as radioisotopes, and they are, as you might have guessed, radioactive. Radioisotopes will undergo a process called radioactive decay. During radioactive decay, the unstable nucleus will spontaneously rearrange itself and throw out particles in the process of becoming more stable. In this example, you can see a carbon-14 nucleus decay by shooting out a negative particle from one of its 8 neutrons. That neutron then becomes a positively charged proton, and now the nucleus has 7 neutrons and 7 protons, and the unstable carbon-14 has now become a stable nitrogen-14. Essentially, by radioactive decay, a carbon has now become a nitrogen, and that is because Ultimately, it's the number of protons that determines the identity of an element. And since this carbon has gained a proton by converting one of its neutrons into a proton, it has now become nitrogen. Radioactive decay can be very dangerous for living things. Those high energy particles that shoot out of an unstable nucleus of a radioactive atom can act like tiny little subatomic bullets. They can damage cells and even molecules like DNA, which is what causes genetic mutations. But as much as they are dangerous, though, radioisotopes can also be useful in biology. One of its uses is for a process called carbon dating. Oh, no, not that. This. Radiocarbon dating works by comparing the amount of different isotopes of carbon in a fossil. So, cosmic radiation in the upper atmosphere keep forming carbon-14 from nitrogen-14. And this keeps the ratio of carbon-12 to carbon-14 in the atmosphere relatively constant. During photosynthesis, plants absorb carbon-12, carbon-13, and carbon-14 in the carbon dioxide that they use to build their cells. They don't absorb those in the same amounts though. There's way more carbon-12, a tiny bit of carbon-13, and an even tiny amount of carbon-14. When an animal is alive though, it consumes that carbon when it eats, and that carbon then becomes part of the animal's body, including its bones. Once the animal dies, it stops consuming carbon, and the amount of carbon-14 that was absorbed into the bones will start to go down as the carbon-14 continues to decay, turning into nitrogen. But that carbon-14 is not replenished because, well, dead animals don't eat. So carbon-12, on the other hand, does not decay, and its amount does not diminish at the same rate as the amount of carbon-14 is diminishing, which is decaying. So scientists know that the half-life of carbon-14 is 5,730 years. That means that they know that it takes 5,730 years for the amount of carbon-14 in a fossil to be reduced by half due to radioactive decay. So by measuring the amount of carbon-14 left in a fossil bone and comparing it to the amount of carbon-12 in the bone, scientists can calculate how long ago the animal died and essentially stopped adding carbon-14 to its bones. Another use for radioactive decay is in medicine. Radioisotopes are often used to trace blood flow or to trace certain chemicals in the body. Radioactive decay can actually be detected by machines like PET scanners. So if atoms of radioisotopes are introduced into a patient's body, like for example by drinking it or by injection or IV, then doctors can follow the path of that tracer or the activity of body parts that use the atom that contains the tracer. So for example, the thyroid gland is the only body part that uses the element iodine. Radioactive elements have the same chemical properties as the non-radioactive elements. So when a patient is given radioactive iodine, the radiation accumulates in the thyroid gland just as regular iodine would accumulate in the thyroid gland. But because the radioactive iodine is radioactive, it can actually be used to provide an image of the activity and the shape of the thyroid gland. In this particular example, you can see that this radioscan is for an overactive thyroid gland. So this person has a type of tumor that is called Graves' disease, which is causing something called hyperthyroidism, which means that their thyroid gland is working much harder and at a much faster rate than it normally would. So when radioactive tracers are used, patients do actually become radioactive and stay that way until all the tracer decays. And that is why radioactive tracers tend to have short half-lives, at least the ones that are used inside the patient's body. In this particular case, iodine-131, which is the radioisotope of iodine that is used in medicine, has a half-life of 8 days. So that means that 8 days after the patient receives or goes through the scan, half of the iodine that was introduced into its body would be gone. And 16 days, there will only be a quarter of that amount left, and so on. So now that we've discussed the nucleus of the atom, let's take a closer look to the electrons and to their location. And remember that electrons occupy a specific region in space called energy levels, or shells, or orbitals. And the electrons of an atom have potential energy because of how they are arranged in relation to the nucleus. So remember that opposite charges attract. So the negatively charged electrons are attracted to the positively charged nucleus. And because of this attraction, it takes energy to move an electron farther away from the nucleus. When energy is added to an atom, the electrons actually absorb the energy and move up an energy level. Almost immediately, though, the excited electron gives up that extra energy and moves right back down to its original energy level. But in the process, it releases that energy it absorbed, usually in the form of light or heat. So if you get a chance to perform a flame test lab in grade 9 science or in chemistry class, that is what is happening there. When elements are heated, they absorb energy and electrons jump up to a higher energy level, but quickly return to their original energy level and release that energy, and usually in this particular case as lights of different colors when those electrons return. So you can buy color changing logs that take advantage of this phenomenon, and the logs are just infused with different elements that burn in different colors. It is also what makes fireworks emit different colors of light as they burn. So how are electrons arranged in these energy levels or shells? Well, shells can only hold a certain number of electrons. The first shell can only hold up to 2 electrons, and the 2 shells that come after it can hold up to 8 electrons each. There are rules for the shells that follow that we will not get into for this course, but you probably will in your chemistry courses. So the outermost shell of an atom is what we call the valence shell, and the electrons found on it are what we call valence electrons. The main goal of all atoms is stability. And stability means having a valence shell that is filled with as many electrons as it can fit. Because the valence electrons are the ones exposed to other atoms, they can be shared or transferred between atoms as the atoms try to achieve that full valence shell that it needs to be stable. Basically, that's what happens during chemical reactions. This is something that we will discuss next lesson when we talk about the formation of compounds. So let's take a look at oxygen and neon. Both have 2 electron shells, but oxygen has 6 electrons in its valence shell, while neon has 8 electrons in it. The maximum number of electrons that can fit on that valence shell is 8, so neon is stable, while oxygen is not, because it still needs 2 electrons to fill its valence shell. And that makes sense. Neon is a noble gas. It is completely unreactive. Because they don't undergo chemical reactions, noble gases like neon have many industrial uses. For example, they are sometimes used in food packaging because they don't react with food. On the other hand, oxygen is very reactive. As a matter of fact, the gases that are used in food bags are basically there to remove any exposure to oxygen, which causes a chemical reaction called oxidation. Sometimes the gases that are added into those bags are noble gases. More often than that, it's just nitrogen, though. On the other hand, if you let the food be exposed to oxygen, it will turn brown usually. That's basically due to a chemical reaction that happens between the food and oxygen. In chemistry, we often use diagrams called Bohr diagrams to show the electron distribution for different elements. Looking at Bohr diagrams of the first 20 elements in the periodic table can help us understand a lot of the chemical behaviors of atoms of different elements. In the periodic table, you can see that elements on each row or period have the same number of shells, with the number of electrons on that shell increasing as we move from left to right. Now, if we look at the columns of the periodic table, which we call groups or families, you will find that the number of shells increases as you move from the top to the bottom. But the number of valence electrons on that last shell is the same for each group, which means that they have similar chemical behavior. For example, the most reactive metals have just one valence electron. They are group 1, the alkali metals. That includes lithium, sodium, or potassium. But even though it is a gas, hydrogen, with also one valence electron and part of that same group or family, tends to behave very similar to the alkali metals. In short, the electron configuration can help us explain the trends that we find amongst elements, like their reactivity or whether they're likely to form covalent or ionic bond. And this is again a topic that we will explore next lesson. So Bohr diagrams are kind of a pain to draw. And really, the valence electrons are the most important for producing an element's chemical behavior. So chemists and biologists tend to use Lewis dot diagrams to represent the paired and unpaired valence electrons in an atom. There are a few simple rules that we need to follow to draw a Lewis dot diagram. First, you draw the atom's chemical symbol. And next, you need to know the number of valence electrons you're going to add. To do this, you can check the periodic table of elements and just count from left to right. So for carbon, there are 1, 2, 3, 4 valence electrons. And so now that you know the number of valence electrons, now we can just start drawing the dots around the symbol. And so we will start moving around from the top in this particular case, and we will put one dot on each side of the symbol, and that's 4 valence electrons, 1 dot per valence electron, and that's it. That's the Lewis dot diagram for carbon. Now, let's take a look at chlorine. There's the symbol. Chlorine has 1, 2, 3, 4, 5, 6, 7 valence electron. And so we will add the 4 as we did before with carbon. But now we still need to add 3 more. So we have 1, 2, 3, 4. Now we're just going to have to start pairing the electrons. So 5, 6, 7. And there you go, a Lewis dot diagram for chlorine. So that's the end of this video lesson. But you're not done yet. At least you're not done learning yet. Because it is never enough to just read or to listen to a lesson or even to watch a video. To really learn and understand a new topic or a new skill, you need to interact with the material. That means actively thinking and problem solving. So every lesson in this course will end with a lesson assignment. And these will be graded and will be found on D2L, so you can access those there. So today's assignment is just a lesson quiz on the content that was covered in this lesson. Most lessons will also have a challenge activity. These are not for marks, but can help improve your understanding of the lesson topics. And along with every lesson, I also always link a few extra videos that either expand on the topics covered or present the information in a different way. Watching these videos is completely up to you and your learning needs. So please complete today's lesson activities and don't hesitate to ask questions if you need anything clarified. Talk to you soon.